promising breakthrough in cancer research. To see life at its beginning is to witness the energy which drives it, the power of cells to divide and multiply, propelling life into the world.
Seek professional help.
But even in growth, death plays its part. Cells in the embryo's hands die off in order to sculpt the fingers.
Seek professional help.
The seeming stability of life scientists are just beginning to realize is only possible by grace of a dynamic balance between new life and death.
Seek professional help.
For beneath the surface of this miracle, tissues replenish themselves, and old cells die as new ones take their place.
Seek professional help.
The true miracle of life is in this balanced and willing exchange of old life for new. Death is not the enemy of life, but its guardian, savior, and companion.
I'm a pathologist. Pathologists are trained to think about how cells know when to die. As a young medical student, this was the one area in my textbooks that actually didn't make sense. It looked as if death was something which involved just running down like a clockwork mechanism. I didn't believe that.
Good afternoon, ladies and gentlemen. We have with us today Professor Willey, who is in research in Edinburgh. Thank you very much.
This is a story about a pathologist whose conviction ran counter to prevailing ideas for nearly 20 years.
Nice to see you again.
A story about that kind of British science which has been kept alive by little more than faith, hope and charity.
And I'm not going to do that today because I think it's too easy to present the sort of science which your work supports as if it was a kind of conveyor belt.
He's a hero. There's no question he's a hero. I mean, he was in the wilderness for years. I mean, for years. I mean, no one was listening. He had trouble getting support for his research. I suspect he had trouble publishing his papers, and he stuck to it. And he was studying something that turned out to be fundamentally important. I mean, really important. And no one was listening. So it's given to very few to have this kind of experience, to see what you've been doing come right to the center of the subject.
And whether you ought to put it aside, or whether you ought to follow it with all you've got. And most of us don't like to confess to the things that happened almost by accident. But I will tell you in just a few words what happened to me. I was working with a great gentleman and a good scientist and a great leader, Sir Alastair Currie.
The health service started as it must.
Professor Currie, another pathologist, introduced Willey to a phenomenon he had first seen in his lab in 1948. Whilst performing an autopsy, he had seen a form of skin cancer called a double squamous carcinoma, which defied conventional wisdom. Somehow these cancer cells, instead of multiplying out of control, had spontaneously withered and died, and the cancer regressed.
In the late 40s, I did the autopsy on that patient, and that was how I began to become interested in this self-healing squamous cell carcinoma of skin. The tumours looked like the ordinary single squamous cell carcinoma of skin, but these were multiple and they regressed spontaneously. Now, I looked at some of the single ones after that and found that in areas within the single squamous cell carcinoma, the normal kind of one, of tumour, that there were areas similar, little areas of regression. And why should that happen?
Even today, sufferers from this same rare inherited form of cancer, like Mrs. Lambie, don't understand why it should regress by itself. She had had and survived this skin cancer over 50 times before she even realized what it was.
It looks a bit like a spot. They can grow to sort of thumbnail size, but they will eventually heal themselves. I never think of it, ever, as being cancer. But I notice it is. Under the microscope, yes.
Wiley began to look more closely, with the help of pathologist John Kerr, at all sorts of tissues for where, why and how cells died.
We began to see tiny little screwed up bodies. The ones that are going to die begin to move about in a rather odd way, and then catastrophe comes over the cell and it shrinks down, and then these bodies are wrapped up by the adjacent cells that eat them. We thought that if cells in a variety of different circumstances show exactly the same structural changes as they die, it's likely there is a common mechanism underlying that sort of death.
And they did find the same pattern of cell death wherever they looked. Such a neat and organized death must, they thought, not be just a random winding down, but a controlled and active program. This kind of orderly death had been observed by embryologists for over a century. They knew it was vital in metamorphosis and growth, but had never considered that it was important beyond development. But Wiley, Currie and Kerr could not believe that what they were seeing was not of greater significance. They began to suspect that somehow a death program could distinguish between healthy cells and ones which needed to be singled out and destroyed. Could the skin cancer that Currie had first seen have been cured by a natural healing process?
Two sets of people. Yeah. And instantly this idea began to develop in his mind and also in mine that death is usually looked at as an enemy. But in terms of tissues, death is a great healer. You like to think when you're stumbling into something, these cells are dying, these are cancer cells and they can die by the suicide pathway. Wouldn't it be intriguing if we could find out what that pathway was and switch it on? You know, we thought these things 20 years ago.
We did think that, and suggested that it was apparently triggered by internal stimuli, which set the cell off on the path of self-destruction. In other words, the cell committed suicide. And we thought that we needed to give it a name. So I spoke to my friend, the Professor of Greek in Aberdeen, and I met the Professor of Latin also, and told each I was asking the other for a name that would fit this phenomenon, having described it to him. The Professor of Greek came up with Apoptosis, that's ancient Greek. And in the original paper, it said the word apoptosis is used in Greek to describe the dropping off or falling off of petals from flowers or leaves from trees.
In effect, the pathologists were suggesting that biologists had failed to recognize a fundamental mechanism of nature, how cells can commit suicide. Few were ready to accept that, or that any such mechanism even existed. Why should cells need a program in order to simply stop living? Their speculations about the fundamental importance of apoptosis and its link with cancer were met with indifferent silence, and their paper was hardly ever referred to.
Well, maybe it was our fault. Maybe we didn't make the case strongly enough. But we thought we had good reason for speculating, good cause for speculating.
We were not in a position to convince our colleagues at that stage that this was anything more than words and faith and hope. At that point we got stuck. We didn't have the tools to explore this any further. It was like very frustrating. It was like finding a few bits of a jigsaw puzzle and seeing that it was going to be a fantastic picture and a great big puzzle. You know, you can sort of see by the scale it was going to be a huge picture with glowing colours. But we just had a few bits and we couldn't seem to take it any further. And I went through a period of great discouragement at that time because I seemed to have got stuck.
Dr. Rob Newbold,
The most exciting thing was
cancer
that
research in London, is one of the biologists
oncogene,
now
which we
trying
knew
to
was
find
involved
out
in
something.
By the 80s, their ideas seemed even less fashionable. The genetic revolution was taking place. Among the first genes deciphered were genes not for death, but for growth. It was these genes which seemed to offer a breakthrough in understanding cancer.
The gene from the normal cell had been stolen by the virus and turned into an oncogene. This trial of Mick, one of
The new idea was that the genes which normally regulated how cells divide became permanently switched on. The cells stuck in growth overdrive. Powered by these ideas, cancer research itself became a growth industry. This is what caught people's imagination, not cell death.
Well, at that time, I think one of the reasons why there wasn't much interest paid to apoptosis, because the fashionable bandwagon then was the area of cell growth, cell proliferation, growth factors, and most people were jumping on that bandwagon. Our bandwagon, if we can call it that, in our bandwagon we very little company. We were there on our own, more or less.
I didn't want to give up doing research. I'd been reared to think of pathology as involving research. I didn't want to give up studying apoptosis. And of course you think, I was involved in this at the beginning, why can't I go on with it?
But he couldn't, because in 1984, his funding for apoptosis was withdrawn.
We mustn't try and give the wrong impression of the sort of stalwart chap who knows where he's going, beaten back by external events. It wasn't like that at all. It was a kind of tentative exploration, not knowing if you were even going down the right corridor or had the right tools. It was much more like that. I remember at that point saying I must really do something else.
Although reluctant to abandon apoptosis, he was fed up not getting anywhere. And so he left Currie's lab to learn the new techniques of growth gene research. But almost as soon as he began his new career, experimenting with growth genes, he once again observed something about them which others had passed over, but which bothered him. In some of his experiments, these cells being driven by the growth gene Mick were not just dividing quickly, but collapsing and dying, appearing as small white blobs.
The first thing we began to see was apoptosis in the cells that were being driven by the growth genes. That was a very important finding to me, because this was when I thought maybe we weren't going to be able to take apoptosis any further.
Wiley thought that the mechanism telling the cells to divide might also be able to tell them to die, that the same genes, the so-called growth genes, might control death as well.
And it turns out that there are genes that drive cancer cells, and they're rogue genes. They're genes that would normally be like the accelerator pedal on a car, but they've got jammed on. And everybody was thinking about these genes as if they were controlling the process of cell production. But because we had thought about apoptosis, we immediately thought, do these genes also control the process of cell death?
Something he proposed in a meeting in 1987, but once again his ideas were far ahead of his ability to prove them. He still had no proof that a gene for death existed. Unbeknown to Wiley, however, the worm was finally turning. And the worm in question was a microscopic nematode, possibly the most interesting worm in the world.
I really went to work on the worm with the goal of understanding information processing in the nervous system. As I decided, rather than studying a human brain, I would try to find a nervous system that was simpler. And the worm actually had the simplest nervous system going in biology. At this point, we know, for example, its nervous system contains 302 cells. That's as opposed to something like 1 million cells that we have in our brains. So 302 I thought I could count to, I thought we could understand maybe at some level.
What makes nematodes so attractive is that they are simple and identical. And unlike humans, almost everything about a nematode is directly controlled by its genes. Which means that when you find a worm with cells missing, or doing the wrong things, it's relatively simple to pin down which gene is responsible.
So in studying the development of the worm, we were watching its development cell by cell. And what we found was that a number of cells would die. They would die as a normal part of development, and if we looked in a second worm, the same cells that had died in the first worm would die in the second. It was clear that these deaths were in some way programmed into the development of the organism.
You can see one right there. You can tell that this is a corpse because it's small, it's round, and it jumps right at you. I knew that cell would be there, and it's going to be there the next time around. It's going to be in that animal. It's going to be there at the same time in development. Here, and every single animal we can look at.
I was so ignorant about developmental biology that I probably didn't know enough to be surprised by the fact that cells were dying. Some cells were becoming nerve cells, other cells were dying. So be it. What could I say?
The worm people decided, however, that the worm was trying to tell them something, since the deaths did follow a pattern. Where there's a pattern, they realized, there should be a gene. All they needed to help them isolate any death genes was a worm where that pattern was irregular. But at 1 mutant in 10,000, it was an eye-straining business.
It's exciting. The one you find actually is extremely exciting. You say, yes, put it away. There are actually a lot of false positives also.
We basically looked for mutants in which patterns of dying cells were perturbed. Too many dying cells, too few dying cells. And we found a few mutant animals that were abnormal in patterns of cell death in that way. Amongst those mutant animals were mutations in the gene SED3. And SED3 was a discovery and an excitement.
How, they wondered, would a worm without the death gene they called SED3 cope with life?
SED3 mutants have no cell death whatsoever. So, again, the first stages look exactly the same. We have the cells that multiply. So at this stage, in the normal animal, we can see maybe a dozen corpses around. Here, 0. Except for that, these animals are perfectly normal.
What that said was, first, that there is a gene that functions in the cell death process. That means there's a biology that is a biology of cell death.
So every cell had its killer death gene, SED3. The first hint that cell death in worms, at least, really was genetically controlled. It was a huge leap forward for worms, but a slightly smaller advance for humanity, since SED3 had no known equivalent in humans. Meanwhile, a busy team was working on the life of brain cells. Led by the flamboyant Professor Raff.
Seek professional help.
It will divide 3, 4, 5, 6 times. Drop it.
Their special interest was in how cells communicate with one another and what those messages are for.
That's an individual cell, so that's the clock.
We'd been studying a cell that gives rise to a particular cell type in the brain, the oligodendrocyte. These are important cells. If you don't have them, you die. And they're the cells that get destroyed in multiple sclerosis for reasons that no one understands. But anyway, we'd been studying these cells, trying to figure out what it is that makes them divide and what it is that makes them stop dividing and become oligodendrocytes. And we had proposed that signals are required to get the cells to divide, but no signals are required once the cell stops dividing. However, someone in America had suggested that this was not the case. And so I suggested single-cell experiments where you pick up a single cell and put it alone in a little microwell without any signaling molecules, with no other cells, and let's test this once and for all.
He had expected to find the cells unchanged, but still alive.
When we looked the next day at these individual cells, not only had they not become oligodendrocytes, but they'd all died. Every one of them had died. So that got me thinking about, if these cells need signals from other cells to survive, maybe all of our cells need signals to survive. And so the radical idea is that all of our cells have this program. Not only do they have the program, but they really want to kill themselves. And the only reason they're not is that other cells are telling them all the time not to do so. That means that if our cells stopped communicating, all of our cells would die within a day. I came back from a series of meetings where this idea had sort of been brewing in my mind, and talked to the people in the lab and said, you know, if I were you guys, or girls, I'd stop what I was doing and I'd start working on cell death. It seemed a bit of a crackpot idea, and there wasn't very much evidence to suggest it was right. I was only kidding, in a way, but none of them did it. So I did it. So I started working on it.
When I got here, he was just starting to formulate what I thought, and I think other people in the lab thought was an incredibly bold, imaginative idea that, I mean, extrapolating from what at the time was only a small amount of data, he began to think maybe this is a general property of all cells. I mean, just imagine postulating that all cells are continuously trying to kill themselves and that they have to be continuously signaled not to do so. I mean, not just some cells, but every cell in your body is trying to commit suicide all the time. And I mean, I didn't believe it. I definitely didn't believe it. I mean, only recently have I started to warm up to it.
Martin walked in on my first day in the lab and said, I arrived thinking, okay, I'm going to work on the retina. And Martin turned around to me and said, boy, have I got a great project for you. I said, oh yeah? He said, cell death. What's that?
It's such a nice hypothesis, and it makes such good logical sense. It's going to be very interesting if it's not the case, because...
So the question was, is this situation where cells need signals from other cells to live a universal one? Do all cells require this? Well, it turns out you couldn't really test that directly because there's hundreds of cell types and it's unrealistic to try to do them all. So what we decided to do was to go for a place where there's only one cell type in a tissue. If ever there's a place where cells should not need signals from other cell types, these should be the places. And there's two places where that situation exists. One is cartilage and the other is lens. In the lens of the eye, there's only one cell type. And to our great surprise, they lived forever, basically, without any signals from other cell types. However, if you now drop the density of the cells in the culture down so that the cells are separated by large distances from their neighbors, now the cells died, and they died by typical programmed cell death. They were killing themselves. So it turns out these cells don't need signals from other types of cells, but they seem to need signals from other lens cells in order to survive. So our view is that if it's true in the lens, then it's going to be true everywhere for all cells.
According to Raff, all cells want to die, but are only allowed to do so by their neighbors if they become superfluous, damaged, or dangerous. In other words, the body's health is controlled not by life, but by death as a fail-safe mechanism.
I think it's brilliant. I think it's a direct extension of the notion that cell death is a programmed phenomenon. Maybe the dawn had come, I don't know. I think that obviously there was a climate of opinion changing, but he was part of the changing climate of opinion, so it's very difficult to say which came first.
Ideas play many different roles in science. One of them is to put your work into a context that people can understand and think, hey, this is interesting, even if it's wrong. If people pay attention and say, hey, this is interesting, and I can understand this, that's very important. Now, if you're wrong all the time, this is not so good.
Every single new postdoc that's come into the lab in the last 3 years has picked a different tissue, a different cell, and no one yet has been able to find a cell that doesn't require continuous signaling. He's offered a sizable reward to anyone who can find a cell type that doesn't need to be continuously signaled to survive. So I was pretty sure that would be no problem.
This sizable award, by the way, is open to any of you. I mean, this is, you know, £1,000 to anybody who can find a cell that can live without signals from other cells. International competition.
I think they're two really different styles. I mean, I really respect Willey, and personally I enjoy very much being a scientist like Willey. He has really, really strong capacity to observe. By looking, by knowing something, he's come out with this very important distinction between accidental death and suicide. What Martin's done is he is someone who likes to go for key issues, so he leaps around biology and says, you know, what's interesting, what's good here, what's good there. And when he arrives on something that he thinks is really good, he just goes straight for the jugular.
So you could say about Andrew, I guess, that what he was doing was crucially important, but he failed to catch the imagination of fellow scientists. Now, I suspect he may have just been too early, but it's possible that it's because he didn't put it into a large enough context.
Some scientists are already excited by Raff's idea that all cells are primed to die. They think that apoptosis may be the key to how the body clears away unwanted cells and keeps itself healthy. For Eileen Quinn, this self-cleansing process went almost fatally wrong. When she developed a lung infection, her body responded normally, sending cells called neutrophils to her lungs to attack the infection, cells which then normally clear away. But in her case, they didn't, and her lungs began to fill up with these cells, drowning her.
It's literally like trying to inhale through a straw. You just can't get sufficient air into your lungs to breathe properly, and it's very, very frightening. I was in intensive care with this oxygen mask on, and I was still screaming, I can't breathe, I can't breathe.
These are the lining of your lung again. Yeah. All these little blobs are neutrophils. Neutrophils are there to protect you against infection.
Chris Hazlitt, who worked with Wiley as a graduate student, now specializes in respiratory problems.
So it's almost like an autoimmune thing, is it?
It was in 1982 that he started to think that cases like Eileen's were caused by apoptosis not working, an idea that came to him when he was asked to give a lecture.
In other words, I thought back to my pathology days and thought, well, maybe I should talk on apoptosis. What's happened to it? It had been almost 10 years. What's happened to it? So I looked at the literature and I found Andrew Wiley's new article in Nature and his review article. And I started reading the review article for this presentation. And as I was reading it about cells being cleared without causing any local tissue injury in embryology and in thymus involution, the penny dropped. You know, it was the closest I'll ever come to a eureka moment. You know, I thought this was it. I didn't have any data for it because I hadn't actually looked at the cells. So I designed very rapidly this experiment and rushed to this experiment with the hope that these cells would look apoptotic. And I nearly fell off my laboratory stool when I saw that these cells were apoptotic. So immediately I picked up the phone to Andrew and said, how do I prove it?
And this very manic voice from Denver came on the phone. It's Chris Hazlitt phoning from Denver. And he was describing what he had seen. Then when he sent the pictures, I realized why he was so excited, because they were beautiful pictures. And there in this central cell of human inflammation, here was once again apoptosis.
Hazlitt's idea was that Eileen's cells had not cleared because they had failed to go through apoptosis. Yet at the very last minute it did happen, and she survived. Cell death was the vital signal for her scavenger cells to clear away the unwanted cells that were drowning her. Hazlitt is now convinced that apoptosis is the body's own healer, which could be used as a powerful natural treatment for diseases like asthma.
No.
Meanwhile, back in Boston, the worms had moved into high gear. They had discovered another mutant death worm, and it was the strangest one yet. Missing a gene they called SED9, the worm would shrivel and die before it was even born. Without the SED9 gene, cells which normally lived died instead.
In fact, it looks like most, maybe even all cells in the animal, when SED9 is turned off, undergo programmed cell death. As if SED9 is protecting every cell in the living animal from dying by programmed cell death.
So the question then was, all right, so here we have a gene that protects you from dying, you know, as a life gene, the gene of life. I mean, cell death in a worm, who cares? The question is there anything in humans that looks like this and is also very important for humans?
First, they had to decode the gene. Then send this information off to the database of known human genes.
what we found is that there was a mammalian gene which seemed to be involved in regulating cell death that looked like SEM9. It's exciting because that made us believe that the way worm cells die and the way human cells die is at the bottom going to be the same. That basically that the whole thing was conserved, that we're looking here at, it doesn't matter if you're a nematode cell, if you're a human cell, you have the same program in you that can kill you if need be. And that what we're doing in the C. elegans is going to be directly relevant to human disease. So it basically justified me, spending all my time working on this little critter that swims on an agar plate, whereas my colleagues were working on cancer genes and important things.
Well, SED9 was very exciting. I mean, the whole world community was excited about that. People were phoning up saying, what does this really mean for cell death as a whole? And this sort of grandiose question. Again, I think it's one of these issues that sort of expands as you get inside it. Science is a bit like potholing, a sport I don't indulge in. But you see a hole and you go through it, and it may just be a little blind hole, or it may open into a great cavern with glittering stalactites.
So now they had one gene telling cells to die, and another saying, don't die. And that savior gene they knew was implicated in some human cancers. 20 years on, there was finally enough evidence to begin to confirm Wiley's initial conviction that apoptosis and cancer were intimately linked, a link that explains cancer cases that had seemed untreatable, cases such as Moira Murray's.
And that was when they said that things had gone so far and I ought to choose where I wanted to die. You inevitably begin to grieve for what you think you're going to have to leave. I remember also thinking that I did not want to miss our daughter's weddings. There were 3 of them. And we had a handicapped son, and rightly or wrongly I felt nobody could look after him the way I did.
If cancer were simply cell growth, then her cancer should have been untreatable because it was growing too fast. No matter how many cells they killed, more would grow, and the treatment would kill her faster than her lymph cancer. But she insisted on treatment.
The injection drugs made me desperately sick. I think it almost bumped me off in itself several times, but it never quite did so, which was fortunate for me.
And to everyone's surprise, her tumour died back as quickly as it had grown.
You can see the tumour here. Here's the heart shadow in the diaphragm. And this huge shadow in the upper chest is the shadow of the tumour. I understand it had been quite a fast-growing tumour. Now, after therapy, here is the X-ray of the same lady. The heart shadow. This little knuckle is the normal shape of the aorta leaving the heart, and you can see no trace left of that tumour. It's almost as if you could predict that the process that switches on proliferation might be linked to the process that causes cell death.
Perhaps what saved Mrs. Murray's life was that whatever had accelerated cell division had then accelerated cell death. A neat idea, but again no proof. Yet.
Well, I'm an unwilling convert to the cell death fraternity. I, like most people, reckon that apoptosis was some weird side issue that had very little relevance to what was going on in real cells, in real organisms. We were really looking at one particular gene which promoted cell growth. It was a gene called MYC. It gets that name from a disease, a leukemia that it causes, myelocytic leukemia. It seems to be important in almost all human tumours, but it wasn't clear what it did. So we were doing experiments where we introduced this MYC gene into normal cells and then looked to see what happened when we tried to stop them from growing. You just withdraw the factors that command the cells to divide. Then cells normally just stop. The only thing was that when we came back the next morning, there were actually fewer cells, even though the cells had continued to divide. And the reason for that was there was a sea of dead cells. So we threw that culture out and thought we'd done something wrong. We did the experiment again and again and again. And eventually, even I realized that something was going on that was quite important. And I think that's the point when I suddenly realized that this could be apoptosis. I knew Andrew's name was associated with that, so I got on the telephone to Andrew and called him up.
And then his phone rang, and he said, we've got these cells that we've put CMYC into, and we don't know where they're all going. They're still proliferating, but the number of cells in the bottom of the dish is staying constant, and we get all these little things floating about on the surface. I felt like saying, snap.
Wiley had seen this 5 years earlier when he'd been experimenting with the new growth genes and had proposed that they also control death. It now turned out that the gene MYC lay at the join between growth and death, where it can act as an accelerator for both. And which it does depends, as Raff argued, on the signals the cell receives from other cells. If it gets signals, MYC accelerates growth. Without them, it accelerates death.
And suddenly I started thinking along lines that I never would have considered beforehand, never considered. Why would you build a cell so that whenever you start dividing, you automatically build in a program to kill yourself? Well, I think in thinking about it, what it suggests is that one of the major ways in which we limit cancer from developing within organisms like man is that every time a cell picks up the machinery to divide, it picks up an abort pathway to kill itself. Now that means it's very difficult for a cell to ever become a cancer cell, because the whole process that's driving it to proliferate is also driving it to die. And the only reason that cells don't die, and this is where it fits in with Martin Raff's ideas very, very well, the only reason that cells don't die is because they get other signals telling them actually not to commit suicide.
I think this concept that proliferation and death are linked together, the same gene pushing the cells into the decision-making cockpit, and that gene often being a cancer gene, that in a way offers an explanation why in dramatic cases like Mrs. Mice, the tumour cell was poised to die. It was dividing, it was proliferating, but it was also very ready to die. And the treatment helped it make its decision in favour of death.
The jigsaw puzzle was finally filling out. And so the chase was on to find out what makes cells choose the death wish.
It's a very simple phenomenon that P53 is essential for cell death induced by irradiation.
So just in Katie's new data.
Wiley already had the Edinburgh team working on a cancer gene known to be involved in 80% of human cancers. Called P53, it was thought to be critical to cell proliferation. But Wiley was convinced it would turn out to be a death gene. To see what this gene really did, he had to study what happens in a living animal that has had the P53 gene engineered out of it.
The expectation was, because P53 is so fundamental, was thought to be so fundamental to the way a cell survives and the way a cell turns over, is that animals without P53 would be seriously ill and not survive to birth. Now that would have been useful because you'd have still been able to look at the function of P53. But the big surprise to everybody in the field has been the fact that the animals develop normally to birth. But what does happen with these animals is as they age, they develop tumours at a very high frequency. And what that tells us is what the real function of P53 in these animals is to stop tumours forming. Normally what happens, if you damage the cells so that they carry mutations, then in an animal with a normal P53 is that those cells commit suicide. However, in our animals without any P53, those cells don't commit suicide. They're indestructible. And what we think is happening is that they carry this mutational load, which is generated by the DNA damage, but the cells are indestructible. They keep on living, and ultimately they form a tumour in the animal. It makes the cells indestructible, but it kills the animal.
So P53 is a guardian gene. What P53 does is to monitor damage to cells from, say, smoking or radiation, and decide if the damage can be repaired, or if the cell should commit suicide. If P53 decides the damage is too great, it switches on the death program. But if it's not working, as in cancer cells, cell death can't be activated, and the cell can't react by dying through apoptosis.
Now you will ask, but what has this got to do with patients? Well, the interesting thing is that now we know the levers, and now we know the end point. Some of my colleagues in Edinburgh and indeed all around the world are now working very hard to get the control of these levers by means of drugs.
Up until now, all the therapies that we've tried to invoke to prevent cancer have been targeted at hitting cell proliferation. That is, trying to stop cells from dividing. Now the problem with that is you hit the cancer cells, but you also hit a lot of normal cells in the body that need to divide in order to keep you together, such as your gut and your skin and your immune system. Now, if it emerges that cancer cells are different from normal cells in that they've lost the ability to die, whereas your normal cells still have that ability to die, they just don't die because they're being told not to die by the cells around them. So now the cancer cell would die if you could correct what's gone wrong with it. Now this is very exciting, because if you could go into the cancer cell, correct what's gone wrong, the trapdoor would open underneath and the tumour would just disappear. And you wouldn't have any of the horrible effects, hopefully, that you do with modern cancer therapies. This is so exciting that you can understand why it's grabbed the community by the throat. And when you think most of the diseases that affect us in the Western world are diseases where cell death appears to play some part, cancer where it doesn't happen when it should, and things like neurodegenerative diseases and AIDS where it happens and it shouldn't.
The rush is on to find drugs and gene therapies that will switch on cell death. Professor John Hickman, a pharmacologist, and his partners in potential profit at Zeneca spent millions of pounds trying to stop cells dividing. Now they're among the first to rethink their strategy against cancer.
It was like looking through a mirror and seeing the image completely differently. And we thought about the growth of cancer totally in terms of proliferation. And I think the whole concept of survival was one that I just found astonishing.
No, we didn't understand any of that. And I think what really is exciting about these new discoveries is not only that it presents us with a challenging set of new targets against which we can mount drug hunting programs, and it really is challenging to do that, but also it's provided us with an explanation of the frustrations of the preceding years.
You know, you hit the cell and the cell reacts. And we never thought about that before. We'd always thought, well, you just hit the cell and it will die. If you hit it hard enough, it will die. Of course, all the tumours that we were affecting in man were derived from those organs where cell death is relatively easily engaged. And that's lymphoid tissue, all of the tumours of the blood. Now the epithelial cells, that's the major cancers in man, come from cells which cover our surface, in the lung for example, and in the colon. They're exposed all throughout our lives to damage, and so they've got to survive pretty well. So their threshold of engaging cell suicide in the presence of damage is kind of up here. In the bone marrow, it's down here. So we were effective in treating those tumours in which the cells could die readily. They have a program of dying pretty readily. The tumours that we don't affect are those where that program is not readily engaged. Their threshold is much higher. So when you give a drug, of course you wipe out all the normal tissues that die readily, and the tumour coming from these cells continues to grow because its threshold for committing suicide is that much higher.
This is a biopsy taken from somebody actually on the table at the minute. They're under anaesthetic at the minute. They've been going for a routine anaesthetic, but on opening the abdomen, the surgeons have noticed that there are large lymph nodes. So the question is here, is there a tumour or not? Normally, we get these bits of tissue and we process them and take a slide and look at that down a microscope, which takes about 24 hours. But because this patient is going for a major operative procedure, it's obviously important to find out right away. So what we're going to do with this is actually do a frozen section, and I'm pretty sure that's tumour. Well, I think if this tumour proves to be a lymphoma, we know that if we treat it with anti-cancer drugs, there's a good chance the cells will die. If it's a carcinoma, there's a fair chance that they won't. And obviously if we could get into this system and manipulate cell death, that would offer treatment for a whole group of diseases that presently we can offer really very little indeed.
We're talking about hundreds of thousands of compounds that have to be tested. So this is not something, even with the most modern robotic techniques, it's going to take 5-10 years before we see the products of all this excitement coming through.
We want to move our findings and the findings of others into the clinic. I mean, that's, for me, what is important, that patients with cancer can actually benefit from this. And we find drugs which improve upon really what are very crude poisons at the moment.
I think cell death and therapies related to cell death processes may be amongst the most important advances in medicine in the next years. The power of death as a therapeutic, if you will, makes me believe there's a whole world there that is completely untapped because the world has not appreciated the biology of cell death.
Trendiness. I mean, when enough people, you get a critical mass, people say, well, it must be interesting. I mean, there's now reviews in Cell, and, Joe Bloggs is working on it, must be important. Whereas if it's only Andrew Willey, I mean, how could it possibly be?
Maybe we kept them all back.
Yeah.
Well, I think it's also...
If it was anybody else, Andrew, this field would have exploded in probably 50 years.
I think it's also conservative. Well, I mean, that it's a rather unusual idea.
It is true that in any field, there is a relatively small group of people who are invited to all the meetings, who are asked to write the reviews, who see each other all the time. And I've been fortunate enough in the few fields that I've worked that I was usually in that cluster. So again, Andrew was not.
Many ideas in science lie dormant for a period because they don't seem to fit into a picture, and then they're picked up again much later. What's been a really very happy experience for me is to still be around and still able to contribute. I once heard somebody give a lecture in which he said that some of the contributions are made by people on the edges, but are not in the centre because they bring into a field elements from outside it. Maybe that's where being a pathologist helped me.
Right, well, it's a very great pleasure indeed now to introduce Andrew Willey, who started the whole ball rolling some 20 years ago now.
Well, thank you very much for the invitation.
In October '93, 21 years after the original paper, Willey's bandwagon is finally crowded. And at major international meetings, everyone is listening.
Earlier on in the day between the growth arrests. So that's my little story, and I thought you'd be interested in it. I'm interested to tell you it because meeting you, I realised that the money being spent in my lab was coming from lives like ours, of Scottish people in their own homes. I go back to my lab thinking differently about the way we do our work.
If you'd like a complete script of that programme, please send a cheque for £2 made payable to BSS, to Horizon, Death Wish, PO Box 7, London W3, 6XJ.
Is it possible you might consider bestowing your affections on another man?
Please, can we sell up and leave Middlemarch altogether?
We should be just as penniless elsewhere as we are here. Only it's desirable Dorothea should know that there are reasons why she shouldn't receive him again.
There was no need of any safeguard against me.
Your wealth is safeguard enough.
Middlemarch, Wednesday, 9:30 on BBC2.
Tommy Lee Jones plays an unpredictable ex-convict reunited with a daughter he's never seen in tonight's powerful film, The River Rat, in 10 minutes. Lessons to be learned now on BBC2 as an uneasy alliance forms in funky black shorts.
Allez vous faire foutre. Retournez donc à Paris.
French bastards?
Hey, je suis français.
Oh yeah, mates of yours?
No, but we have met before.
I could have had him, I didn't need you to butt him.
They would have killed you.
Bollocks.
So why did you run?
Fancy the jog, that's all.
You English. You are so economical with the truth.
Listen, Frenchie, I don't need your crap, okay?
Hey, my name is Philippe. And my crap just saved your roast beef ass from a beating.
My name's Ellis. And the day a frog saves me. The greatest goal there ever was. There ever was. The greatest goal. Juggling the ball between both feet. He moved 20 yards like Jesus on water. His feet never touched the ground. He was in the hands of God. Centre back. Lunged at him, but he was already gone. The goalkeeper, arms flapping frantically, feebly, futile. And when